Hello, everyone. I'm Shahadat, and today our presentation on the poster is going to be, which is titled as Causal Discovery of Gene Expression, Clinical and Pathological Variable Implicates a Direct Role of Mitochondrial Dysfunction in Chronic Traumatic Encephalopathy. So here is a little bit background of our study. So CTE, which is Chronic Traumatic Encephalopathy, is a progressive neurodegenerative disorder associated with repeated head impacts, often seen in contact sport athletes and military personnel. While substantial research has shown that many gene expression pathways are altered in the brain of individuals with CTE, it's been difficult to determine whether these molecular changes are causes or the consequences of the disease. In previous studies, our group actually identified strong correlations between gene expression changes related to neuroinflammation, neuronal dysfunction, and the accumulation of tau protein, which is a hallmark pathology of CTE. However, correlation does not reveal causation. So in this study, we sought to move beyond correlation and uncover potential causal relationship between genetic factors, clinical variables, and gene expression in CTE brain. We developed a novel causal discovery algorithm that integrates gene-level principal component analysis with clinical risk and neuropathological variables from postmortem brain tissue.
Okay, hi, my name is Peiyu. Our study is based on a comprehensive data set derived from 221 post-mortem prefrontal cortex sample from human subject maintained by the CTE Brain Center at Boston University. The subjects spanned a complete range of CT pathology. Specifically, this cohort include 149 individuals with a post-mortem diagnosis of CTE spanning the full range of pathological involvement from stage 1 through stage 4 as characterized by the McKee staging criteria. As control groups, the cohort also includes 54 individuals with a history of RHI but no CT pathology and 16 non-athlete, non-RHI individuals. The CTE and RHI controls all experienced repetitive head impacts as a result of violent sports play. We also generated high-throughput RNA sequencing datasets from the prefrontal cortex of those participants, which yields expression counts for about 20,000 unique genes. In parallel, we obtained an extensive clinical dataset for each participant, consisting of 116 variables that encompass demographic, clinical, and quantitative neuropathological metrics. A key characteristic of this clinical dataset is the presence of missing values. For example, data values for the ApoE genotype and total years of sports play are missing for 14.9% and 8.6% of the samples respectively. The integration of this heterogeneous dataset provides a powerful foundation for downstream causal discovery and differential analysis.
I'm Ruo Hong. The goal of our method is to find putative causal relationships between genotype, gene expression, and clinical covariates. Specifically, we sought to identify causal relationships between gene expression, amount of tau pathology quantified using immunohistological staining of AT8, total years of violent sports play, ApoE genotype, three measures of cumulative head injury impact as measured by helmet sensors, and numerical CTE stage 0 through 4, where controls are considered as stage 0. To incorporate gene expression data into our causal modeling framework, we first applied gene set variation analysis on the RNA-seq data using the molecular signature database C2 collection. This allowed us to summarize expression profiles at the pathway level, improving interpretability and reducing gene-level noise. Next, we performed principal component analysis on the GSVA scores to further reduce dimensionality. We intentionally applied GSVA before PCA to preserve pathway-level interpretability in the resulting principal components. To handle missing clinical data, we used multiple imputation, which allows us to retain all samples in the analysis. Specifically, we generated 5 imputations of the clinical covariates that were each used in downstream analysis to account for uncertainty in the missing values. We then combined the GSVA PCA project values and the imputed clinical covariates into a causal discovery algorithm. Specifically, we use the Peter-Clark algorithm on each imputed dataset, using the top 10 GSVA-based principal components and relevant clinical covariates as input. This resulted in a set of causal graphs, one for each imputed dataset, from which we generated a consensus network to identify robust and consistent relationship across imputations. Once we identified principal components with putative causal links to clinical traits, we projected these findings back to the gene level by multiplying the principal components loadings with a gene set membership matrix. This allowed us to pinpoint which genes were the most represented in the causal components. To compare our results, we also did conventional differential expression analysis to understand the directionality and the significance of these putative causal genes.
Hello, my name is Wisdom, and at the end of the day, our causal discovery pipeline produced 5 principal component-based gene set causal graphs, and we can see that PC5 emerged as the consensus driver that links gene expression to tau pathology. In the acyclic semi-directed graph you see in the red arrow here, the algorithm identified known causal relationships between ApoE, AT8, and CTE stage, which is largely determined by the degree and distribution of tau pathology. In addition to these expected causal relationships, the algorithm also identified a robust causal association of PC5 with AT8. This topology was stable in 4 out of our 5 imputations that we did. Showing us that there's high confidence in the signal, and it's not just an artifact of missing data handling. Subsequently, we identified the top loading genes contributing to PC5, and the traditional differential expression analysis that we performed also flagged a few yet causal model highlighted tight cluster. The top rank hits were NDU-FA7, NDU-FB2, COX6B1, ATP5, MF, and others. And all these almost exclusively mitochondrial electron transport complexes, 1, 3, and 5. We then fed the top 200 PC5 genes into gene set enrichment analysis pipeline. And from the bar plots, you can see that there's a clear peak for oxidative phosphorylation and ACE-related mitochondrial processes, even after adjusting for p-values. So in conclusion, these results show that mitochondrial dysfunction could be a putative upstream driver of tau pathology in CTE, suggesting mitochondrial bioenergetics pathway as a possible therapeutic target to intervene in the development of pathology and disease. Thank you.